Okay, good morning, students. Let us begin with our today's lecture. Okay. Before that, I'll just try to share my screen with you. I hope it is visible to you now. Is it visible?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So yesterday, I think we had discussed about the different types of materials which are used for making gels. We have seen agarose gel, then we have seen polyacrylamide gel. Okay, and of course there was one small point which I actually forgot to tell you yesterday. That was about in agarose gel there is one more type of an interesting technique which makes use of not slab gels, but it makes use of column gels. Okay, so what is a column gel? A column gel is a tubular type of a gel. It is also called as a tube gel. Okay, now in this type of electrophoresis, instead of using a slab, yeah, on which we have say multiple wells, and in each well we can add a different sample. Okay, but in a column gel we have a tubular type of a gel on which only one sample can be applied. Okay, so basically what we do is we have open-ended glass tubes. Okay, you can say open from both the sides, and within this tube which we can fill up the acrylamide or sorry agarose gel. Okay, molten agarose gel. Then we keep the tubes for to allow the gel to polymerize. Okay, and after the polymerization, the column the glass column with the gel inside is added is attached to the electrophoresis apparatus. This unfortunately I don't have a diagram here, but next time surely I'll make it a point to show you the diagram or a schematic figure of how the column gel electrophoresis apparatus looks like. Okay, so there are two tanks which contain the buffer, the upper tank and the lower tank, and the two tanks are interconnected by these column gels. Okay, so from the top we have the buffer which is in contact with the tube gel, and from the lower end the tube gel is in contact with the lower tank buffer, and then we start the electric current, obviously from the top to the bottom, that is from the negative electrode to the positive electrode. The sample which is applied on the gel will run through the tube gel, and it will separate into different zones. Okay, at the end of the electrophoresis we can remove the column, okay, from the tube, and then this column is put in a staining solution to stain the protein bands, right? I think I'll try to share just a minute. Huh? I'll try to share a whiteboard so that although I do not have a schematic diagram, I'll try to draw how it looks like. Okay, so we have the upper tank, something like this, and then we have a lower tank. Okay, these two tanks contain the buffer, something like this. Okay, and then these two tanks are connected with the tubes. Okay, so of course multiple such tubes can be put here. We have at least say 6 to 10 tubes, okay, which are interconnecting the two tanks. The negative electrode is connected to the upper tank, and the positive electrode is connected to the lower tank. And then when we start the current, what will happen? Okay, before that we add the sample here. Okay, so suppose this is a glass tube. Okay, this tube is hollow from both the sides. Okay, while when we cast the gel, of course this is put on a stand sort of a thing. okay, so that the lower end actually is closed, and then we put the gel material inside. Okay, the gel, for example, will occupy this much of space. Okay, and then when we allow it to polymerize, the gel will polymerize inside, right? And then there is a, can you see there is a space here? Okay, there is an empty volume here. So in this empty volume we can add the sample. Okay, so now we are adding the sample here, right? After having added the sample, then we take out this tubes from the stand and we put it in the electrophoresis apparatus, right? And then we connect the electrodes and we start the electrophoresis run. Okay, so now what is going to happen? The sample which was applied here, okay, the sample is here, and when we start the current, the different components of the sample will start moving, okay, this way, and they will form zones. Do you understand what I'm saying?
Yes.
The different components will move.
Sir, hello.
Yes, yes.
Sir, Sami Marbaz has been unable to join the lecture because he is having connectivity issues.
Okay. So you can just tell him that I am recording this. Okay.
Yes, sir, I told him.
एक मिनिट, रेकॉर्डिंग आपलं होतंय का नाही मला कळत नाहीये.
Yes, sir, you have started recording.
Yes,
It
yes,
showed
yes.
us.
Yeah, it is.
This is the recording.
Good. So the recorded files will certainly be shared again. I don't know how big the files will be. So as much as possible, I'll try to share them, or maybe when we meet sometime physically, then I can give these these lecture recordings to you. Okay.
Yes, sir.
And if possible, then again tomorrow I'll try to repeat. If he's not comfortable, then I'll repeat this. Okay. So the components get separated in the tube gel, and then after some time, of course, there is a tracking die which tells us how much of the run is complete. And then if we feel that the run is complete, that is the die front should come at least till 3/4 of the distance of the gel. Then we stop the electrophoresis, and then we can remove the tubes from this apparatus. Okay, once the tubes are removed, then the gel is removed from this tube, okay, by either pushing it from one end or by pulling it from the other end. Okay, by applying some kind of a vacuum, we can pull out the gel, and then the gel is put in a tray. I'll show here the tray. Okay, sorry. And in this tray, there is a staining solution in which we put this particular gel. Okay. So gel tube, there may be multiple such gel tubes, and all these tubes, we put it in the staining solution. It may be Kumasi brilliant blue, or it could be maybe silver staining, or any other kind of a stain. And the protein bands which are already separated, they will get stained, and we can visualize the bands after destaining it. Okay, so after staining, we have to destain the gels. Why? Because the complete gel is going to be stained with the dye. We have to remove the dye from the regions where the protein band is not present. Okay, wherever the protein band is present, that will retain the stain, and wherever the protein is not present in the gel, that portion of the gel cannot retain the stain. The gel will be the dye will be released from the gel, and that portion will appear white in color. Okay, or colorless. Is it okay? So coming back to the presentation. Okay. Now this particular technique is useful in a way, because here we can do what is called as activity staining. Okay, activity staining is what? One particular, we can run the same sample in multiple tubes. Okay, same sample can be run, and then one tube can be taken out, it can be stained. One gel tube can be stained, say with Kumasi brilliant blue. The other one we can stain it with some other dye if required. Okay, and then there is an interesting type of a technique which is called as activity staining. So for example, we have an enzyme. Okay, if we have an enzyme which is being purified, okay, there are multiple bands in a single tube corresponding to different components of the sample mixture. Okay, and then one of the component in this mixture is probably an enzyme. Okay, now what we can do is we can put this particular tube in a tray which contains the substrate solution. Okay, a chromogenic substrate for the enzyme. Do you know what is a chromogenic substrate?
Yes, sir.
What is it?
So imparts color.
Yeah, so when this substrate is reacted upon by the enzyme, then the enzyme will convert that substrate into a colored product. Okay, that colored product can be visualized. Okay, so now in this case, we can put a chromogenic substrate, and the band in the tube, okay, the band in the tube which corresponds to the particular enzyme, that band only will react with the substrate. Okay, and it will give you a color that will indicate that the particular band contains the enzyme of interest. Is it okay?
Yes,
So
sir.
this particular technique is called activity staining. Now if the
So
same
for
thing,
example, beta-galactoside is reacted on X-gal.
yes,
Uh,
exactly. So X-gal will show what color after the reaction?
so blue, I think.
Yes, blue color. Okay, so the blue color will
Yes.
indicate, the blue color at a particular region in the tube will indicate that the protein or the enzyme is present in that region, in that zone. Is it
Yes, sir.
okay? Okay, the advantage here is that we don't have to cut the gel. If it was, for example, a slab gel, okay, if it was, for example, polyacrylamide or agarose slab gel, then we would have to probably cut the gel. Okay, and one portion of the gel we would put it in the we would put for activity staining, so that we will look, we can localize where the particular protein is present or where the particular enzyme is present. Okay, and then from the other portion, maybe from corresponding to the in the other well, we may have put the same sample. Okay, so the corresponding portion can be removed from the gel, and then it can be treated or it can be crushed so that the enzyme can be removed from the gel. Okay, but that will take some extra time and extra efforts. Instead of that, if you can use a column gel, then one of the column can be sacrificed, you can say, for the activity staining. Okay, the other column will always be there, and from that other column the corresponding region of the gel can be removed, and the enzyme can be taken out, it can be extracted from that particular gel. Okay, so that is the application of a column gel. Okay, you can do activity staining. It can also be used for preparative application like the one I have already mentioned. We can remove the portion of the column, and the enzyme can be extracted from the particular region. The drawback of this particular technique is that only one sample can be run per tube, okay, or per gel. On the other hand, when we studied the slab gel, we could run multiple samples in the same gel. Okay. So of course, here multiple tubes can be inserted in the same apparatus, and therefore multiple samples can be run. But again, the drawback is that all the columns are different columns, isn't it? So when there are different columns, there may be some difference in the preparation. Okay, and that difference might lead to non-uniform results. Okay, whereas if it was a single slab gel, then under the same conditions of electrophoresis, you can expect uniformity, isn't it? So if you run multiple sample, or rather same sample in different well, then you can expect the similar result, okay, in all the lanes. Okay, do you know what is a lane? What is a lane? Okay, the sample components which are running in from the same well, okay, they form a lane, okay, and in that one lane you can get multiple zones corresponding to different sample components, isn't it? So in a slab gel, there is we can expect uniformity in all the lanes together. That uniformity cannot be actually assured in the column gels, right? Okay, then we move on again to another important technique, and that is called as SDS page. Okay, can you see the slide? Have you
Yes,
heard,
sir.
Yes, sir.
Yes, sir.
have you heard
Yes,
about
sir.
this technique anytime?
Yes, in
SDS
third
and
year,
yes, in third year. Okay, that is sodium dodecyl sulfate, which
dodecyl sulfate.
that is a detergent. Okay, anionic detergent, and this SDS is incorporated into the sample buffer, and then it is run in a normal polyacrylamide gel. So that is called as SDS polyacrylamide gel electrophoresis. Okay. Now here, the idea of using SDS is that being an anionic detergent, it has a very high affinity for the proteins. Okay, and when it reacts with proteins, it actually sort of it encapsulates a protein molecule. Okay, convert it into some structure, maybe a rod-like structure or something. Okay, and this particular protein molecule or protein SDS complex carries a very high negative charge. Okay, because SDS is an anionic detergent, it imparts negative charge to the protein. Okay, now you can imagine that in our sample, okay, our sample contains a mixture of different proteins. These proteins have different properties, okay, different charges, different charge distribution, and they have different molecular weights. Now, when we add SDS to this particular sample, then SDS is going to uniformly coat all the different proteins in the sample. And therefore, all proteins will have a very, very high negative charge. In other words, what we are doing is we are imparting a negative charge or a high negative charge to all the proteins in a particular mixture. And therefore, there are no charge differences in the proteins at all. All proteins are highly negatively charged. Okay, and therefore when we run this kind of electrophoresis, the separation of protein that will happen in electrophoresis will be basically on the basis of their molecular weight. Okay, not the charge, but on the basis of molecular weight. Is it correct? Okay, then another thing that we do is in SDS, the sodium dodecyl sulfate detergent also does something to the protein, and that is it causes the denaturation of the protein. Okay, in the sample buffer we also add what is called as beta mercaptoethanol. Okay, this is another chemical which which is a highly reducing agent. So it reduces the disulfide bridges in the protein structure. Okay, and therefore it denatures the protein. So there are two things that are denaturing the protein. One is the beta mercaptoethanol, and the other is the SDS. So when both of these act together on the protein, what happens is because the disulfide linkages are broken, the protein tertiary structure will be broken down into the the different subunits of the protein. Okay. So what will happen? One single protein, if it is made up of two different polypeptides, then the two polypeptides will be separated from each other, right? Now, if the two polypeptides are having a different molecular weight, then what will happen? In the electrophoretic run, the two polypeptides of the proteins will be separated from each other. Okay?
Yes,
In other words, if you want to characterize a pure protein, okay, just remember what I'm saying. If you want to characterize a pure protein to know whether the protein contains a single type of subunit or multiple different types of subunits, or if it is made of a single subunit or multiple subunits, then you can run SDS page. Okay, SDS page, the SDS and mercaptoethanol, what it will do, the disulfide linkages will be broken, the protein will be broken down into the constituent subunits, and the constituent subunits will run in the electrophoretic gel. Okay. Now, for example, I'll just tell you that antibody molecule contains heavy chains and light chains. You know that, isn't it?
Yes,
Heavy
sir.
chain has molecular weight of say 1 lakh Dalton, and the light chains are have a molecule. Oh, sorry, 1 lakh. Is it correct? 50,000 rather.
Yeah.
50,000 is
So
the
50, 50, 25, and 25.
50,
50,
1 lakh.
50, 25, 25. Isn't it? So now what will happen when we do SDS page? The antibody molecule is going to be broken down into four different parts. Okay, two parts, that is the two heavy chains, and there will be two light chains, right? But what will happen, the two heavy chains will run together because they are almost similar in molecular weight, right? So heavy chains will run similar, and light chains are also going to run at in a similar fashion. Okay, because the light chains also have the same molecular weight, that is 25,000 Dalton. So how many bands you will see? Antibody bands. How many bands you will get?
So 2.
You
2
will get
bands.
two bands only. But there are actually four chains,
Yes.
isn't it? There are
Yes.
actually four chains. So how will you know whether there are four chains or two chains? So that will be on the basis of the molecular weight. See, the complete antibody molecule has a molecular weight of 1,50,000.
50,000.
Okay, complete antibody molecule has molecular weight of 1,50,000. And when you run it on an SDS page, okay, you will get two bands. Okay, and when you run the molecular weight markers for comparison, then you come to know that the two bands of the antibody have a molecular weight corresponding to 25,000 and 50,000, isn't it? So when you add up 25,000 plus 50,000, it becomes 75,000. Okay, but the molecular weight of the native protein, that is complete whole protein, is 1,50,000. So how do you account for this difference? Total protein is 1,50,000, and the two bands together correspond to 25 plus 50,000, that is 75,000. Okay, so definitely each band must be corresponding to different polypeptides, isn't it? One band doesn't mean that it is a single polypeptide, but rather one band is two polypeptides together. So 25,000 plus 25,000 is 50,000, and 50,000 plus 50,000 is 1 lakh, and the total becomes 1,50,000, right? So this is how you can characterize a protein. So SDS page basically is a technique not only to separate the protein components. I have 10 minutes, so in 10 minutes I'll try to wind up this technique, and then maybe if Tikekar sir will allow me, I'll continue, or Tikekar sir wants to take the other lecture. Okay, so I'll try to wind up this particular technique at least, right? So what will happen is your SDS page will be SDS page is used to characterize a protein. That is, you can find out the information whether the particular protein contains a single type of subunit or there are multiple subunits. Subunit means polypeptides, single polypeptide or multiple polypeptides, and whether the polypeptides are of the same type or whether they are of the different type. Okay, so there is a term which we use, that is called homodimer, homopolymer, or heteropolymer. So homopolymer means it is the polypeptides are of the same type, and heteropolymer means the polypeptides are of different types, right? Okay, I won't read the slide, but I'll just try to take out the important things from it. We can, since the SDS page is based upon the molecular size of the proteins, it is also a technique which can be used to find out the molecular weight of either the constituent subunits, that is the polypeptides, or of the whole protein as such. Okay. Okay, I'm not reading all these things, 100 volt, 15 ampere, because most of these things are common to different types of electrophoresis techniques. Okay. But before I close this topic, okay, for the time being, I want you all to understand one thing, that what is the application of SDS page, okay? Or in general, electrophoresis, what are the applications of electrophoresis? Okay, one thing is it can be used to separate the constituents of a mixture, isn't it? If I have a
Sir,
mixture of different proteins, and the different proteins will be separated from each other. That is one application. The other application is to characterize a protein. For example, if you are using SDS page especially, then we are able to find out the molecular weights of the protein. How we find out the molecular weight? We run a marker protein, okay, protein marker in one well. Okay, what is the composition of a protein marker? A protein marker will contain different proteins which have standard molecular weights, okay, that is known molecular weights, okay. And when we run that in one well, in one lane, then what will happen? We can find out, we can get distinct bands corresponding to different molecular weights. Now we are going to compare these bands with the bands which we obtain from our sample, and then we assign the molecular weights to the different you can say components which are separated from the mixture. I'll try to show you one gel which I had prepared some time back. Can you see this? Just a minute. Can you see this?
Yes, sir.
Okay.
Yes, sir.
On the right hand side, okay, my right hand side, okay, that is this particular lane. Can you see this lane? This lane is
Yes,
the
sir.
molecular weight marker lane. You can see very distinct bands, and these bands are corresponding to standard molecular weights. Okay. And then all these other lanes, 1, 2, 3, 4, 5, 6, 7, and 8 lanes are there. Okay. So these 8 lanes are containing the sample which contain mixture of different proteins. Okay. Now why I have used this particular gel was to actually find out whether I am able to purify my protein. So what I have done is using different techniques of protein purification. Can you know? Can you tell me any method of protein purification you have learned earlier?
Paper chromatography.
Huh? Yes.
Paper
Chromatography. Okay, different techniques of chromatography. Of course, we are going to again study them in detail in this particular paper. Okay. Then we can use fractionation. Do you know what are fractionation methods? Salt fractionation, pH fractionation, temperature
No, sir.
fractionation. Okay, so all these techniques you are going to learn in MSc. Okay, so for example, when I say salt fractionation, what I actually do is I add a salt like ammonium sulfate to a protein preparation. Okay, and at a particular concentration. Now at this particular concentration of the salt, one particular type of protein in the mixture will get precipitated. Okay, then I increase the concentration of the salt. When I increase the salt concentration, then another different protein can be precipitated. Now one by one I can remove the precipitates, and then I can treat them as crude preparations. What is a, or we can say partially purified protein preparations. Okay, so one by one component we can separate by precipitation, and then each of the component can be treated as a partially purified protein component. Okay, now when I want to monitor this particular protein purification steps, what I can do is, suppose I have used one particular salt concentration here. Okay, and I have run the electrophoresis to find out whether my particular protein is getting purified. Okay, now in this lane the purification is not very great. Okay, in this lane also the purification is not very good. When I go to this lane, can you see this lane? Most of the proteins are already removed, and I get very few bands here. Here there are multiple bands. Here there are multiple bands. Okay, but here the number of bands are few, and maybe one of the band corresponds to my protein of interest. Okay, then I carry out another method, and then maybe in one particular method I get only a single band in the preparation. So when I get a single band in the preparation, it means that I have been successful in purifying my protein. So what am I using electrophoresis for? I'm using electrophoresis technique here to monitor the progress of my purification. I'll show you another gel here. Fortunately, I have another gel here. Can you see this? Can you see? Okay.
Yes, sir.
So here, these bands are corresponding to the molecular weight marker, and this particular band, they are corresponding to a preparation which was subjected to different methods of protein purification, and I could get a single band here. Okay, I could get a single band here, but this band is rather thick. It may be probably a mixture of different very similar proteins here, but then this band here is a very sharp band. Can you see this? Okay, a single and sharp band indicates that I am successful in my protein purification. Okay, so can you recapitulate the applications of electrophoresis in general now? SDS page, for example. Okay, what are the applications? Who will tell me? One is to find out whether a particular sample is a pure preparation of a particular protein or it is a mixture of different proteins. That is one application. Another application is to monitor the process of protein purification. Okay, whether I am successful in my protein purification, that will be indicated by the SDS page. Any other application? Third application is to find out the molecular
Sir.
weight of a particular protein.
Molecular weight.
Right? Molecular
So
weight.
we can denature the proteins and get a better resolution.
Yeah, that's what in SDS page we are denaturing the protein, but the reason here is to find out whether a particular protein contains different polypeptides or only one polypeptide. Okay, so we are able to, we can use SDS page for characterization of a protein also. That is the fourth application. Is it okay?
Sir, also protein causes foaming in the buffer, I think, and SDS is added to prevent the foaming or
No,
anti-foaming agent.
in fact, what you're saying is just the opposite. SDS actually is a detergent, and when you add a detergent and shake it, it will cause a lot of foaming. Okay. Yeah, in fact, SDS results in foaming, and therefore you are not supposed to vigorously shake your samples when you are using SDS. Okay. So, any questions? Shamim had some problem with the internet. Shamim, are you comfortable? Have you understood what I was telling?
Yes, sir. Yes, sir.
Okay. Okay.
I already let in this lecture.
Yeah. So we were just discussing only one technique, that is SDS page. In the other lecture,
Yes, sir.
we will go for other interesting technique. Okay.